All right, everybody, welcome. My name is Brian. I am the Vice President of the Colorado State Beekeeping Association, as well as the Sustainability Lead. We are excited to have Dr. Frank Rinkevich from the USDA lab in Baton Rouge tonight, talking to us for our May Sustainability Chat. Dr. Rinkevich, how are you doing tonight?
Doing very good. Doing very good. Got my boat ready for tomorrow morning and excited to wake up early and try to do some fishing before work. So, I mean,
Nice.
good
Right
time.
on.
Yeah.
Well, are you all ready with your presentation?
Yeah, let me get this up and ready to share. And
How long have you been at the Bee Lab at the USDA?
I've been there for since 2016, so 9 years.
Okay.
Yeah. In that 9 years, we've done a lot of research, and I'm going to talk about some of that research tonight. Um, so I've been working at the lab for, like I said, 9 years now, and hopefully for a lot longer because there's a lot more work to do with honeybees. As we all have heard last year, there were some people that experienced catastrophic colony losses. And it's one of those things that still bothers me about why we're still having these things happen and how widespread it is. And when I first started at the lab, some of the first experiments that we did were these longitudinal studies that usually 1 or 2 years. And you can see on the slide in the picture here, this is what I think about when I think of science. It has to be in the field with beekeepers getting real world data. And when we were doing these longitudinal studies, we would collect massive amounts of data from these colonies to find out why some survived and why some died. And when we would spend all this time and all this money looking at the effects of, parasites, pesticides, viruses, forage, gut microbiota, gene expression, if there's a thing you can measure in honeybees, we measured it for these experiments. And when we did the statistics said it's Varroa mites. And not just a little bit, but 70% of the reason why bees died in all these experiments I was doing was because of Varroa. So that immediately sold me on the idea that I have to study, if I want to help the honeybees, I have to study varroa mites. And so right now my research has two major foci, foci, plural focus. And the other one I work on is Amitraz resistance in Varroa. That's pretty much going to be my career. I don't think that's going away or anything. So that's one of the feet I have my uh, camps in. Then the other one I'm going to talk to you tonight about is breeding Varroa resistant stocks and their future beekeeping. This is a project that started about in the late 90s at the lab, and I'm just the most recent torchbearer of this long-term breeding program. And breeding is very important, especially when we're talking about honeybees, but not just honeybees. It's important for all of agriculture because actually agriculture is just the end result of selective breeding. So when we think about this in agriculture, on the left-hand side here, you could see that there's this progressive picture of corn. So corn didn't show up on this planet, this big giant lump with knobs all over it that we see here on the right-hand side, but it started off as this small piece of small collection of seeds on some grain, on some grass. And then over time, people selected it for larger and bigger stalks until we eventually get the industrial product that we have today. There are many, many, many varieties of corn, but we only grow a few of them here in the United States. So corn, just this phenomenally productive plant, is the result of selective breeding over thousands of years. But also, too, the things that we like to eat, these cruciferous vegetables, things like broccoli and cauliflower and kale and kohlrabi, these kinds of things, they're all actually different varieties of the same plant. You can cross these and interbreed them. And these are just varieties that were derived from selection on certain characteristics of that plant. So if you look at the top here, if you select for the size of the terminal bud, that's how you could select for cabbage. Similarly, if you're looking at the flower cluster, that's how you would select for cauliflower. Well, one of my favorite things that's been selected from this wild mustard plant are Brussels sprouts. So this is very important that we have this variety. But what's interesting is that scientists at the USDA bred that stinkiness out of Brussels sprouts. I don't know if you remember a long time ago, but Brussels sprouts used to be quite funky. But we just improved them with selective breeding to get rid of those sulfur compounds. And that's why they're on the menus of most restaurants nowadays. So it's not just plants, but we could also do this with animals as well. And if you have cattle or know people that raise cattle, you know that there's different breeds for the different kinds of products that you want from that animal. So on the top, if we're looking at milk, we could think about it in two different kinds. We could think about quantity. You know, when you're thinking about quantities of milk, we typically think about Holsteins. Holsteins are incredibly productive milk producers. Some of the most largest volumes are from Holsteins, the black and white ones. But on the right-hand side, Jersey cattles have very high quality milk. So they typically have higher butterfat content, higher protein, and a number of other characteristics that give the butter that's made from Jersey breeds a lot more different characteristics. And it's really important if you like to bake, because if it calls for European butter, you better use European butter because it's a very different product than butter that we have in the States made with lower quality milk. Similarly, if you're producing beef, Black Angus are the standard of beef production, especially here in the United States. They're very adaptable, very productive, and you could do hybrid crosses to get them even more productive and things like that. They're a very prodigious beef producer. But if you want really high quality, you might think about something on the right, like the Wagyu cattle over in Japan, which are known for their marbling and extensive flavor. So it depends on what you want. Quality, quantity, mix of both. Choose the right animal for the production you're seeking. Well, honeybees are really no different. If you can measure it in honeybees, you could breed for it. And this has been happening for about 10,000 years now with different characteristics. We just have better ways to track it nowadays. So with honeybees, we could select for things like high and even honey production. You see on the left how even those frames are and just packed right to the edges with honey. Very consistent product. We could also select for color of bees. You know, some people like light colored bees, like this Cordovan queen you can see here in the middle. Some people like very dark bees. That's something that you could actually breed for quite easily. On the upper right-hand side, we typically breed for bees that have huge population sizes. So this is something that is very good because as soon as that nectar and pollen come out, they could build up their colonies, and especially in preparation for almonds. So that's definitely a desirable characteristic. One of the interesting breeding programs that has been facilitated scientifically has been Marla Spivak's hygienic testing for bees. You can see here in the bottom center here, this is the freeze-kill brood assay that measures how hygienic these bees are at removing brood, in this example, that were killed with liquid nitrogen, but it's a surrogate measure for how they can deal with larvae and pupae that are infected with bacterial diseases. And then finally, something like pollen hoarding on the right. Rob Page has selected this behavior in bees many different times by just finding the ones that store the most pollen and continuously breeding from that. So if you can measure it in honeybees, you could select it and breed from it. It's a little challenging. I'll tell you about some of those challenges to breeding honeybees a little bit later, but this is something that we could definitely do with bees. And this is actually the mission of the lab that I'm currently employed at, is that I work at the Honeybee Breeding Genetics and Physiology Lab here in Baton Rouge. But I almost don't work on honeybees at all. I mostly study Varroa mites. So we should pretty much change the name of this lab to the Varroa lab, because I work on Varroa in many different ways, and then almost everybody at our lab, some part of the research centers on Varroa mites. And the reason why we study Varroa a lot is because beekeepers tell us that Varroa is the number one cause of colony losses. If you look in the upper left-hand side, Varroa, most our people are saying that Varroa are the reason why their colonies died. And this is supported with other research and other surveys, and kind of contemporarily, especially right now, this is what people are saying are affecting their bees. And then finally, too, we know from scientific studies that Varroa cause a lot of damage from feeding on bees. In the upper right-hand picture here, you can see where these dots are. This is where Varroa feeds on the adult bees, and this causes damage to the bee, especially as an adult. That feeding also allows them to transmit viruses. You can see in the bottom left-hand picture that Varroa on the bee here on the abdomen. You can see deformed wing virus is one of the many viruses that Varroa transmits. So it not only damages them from feeding, but also to the viruses. Well, this is bad, and so people, especially beekeepers, would apply miticides, and in the case here, Apivar. And Apivar worked really great. It contained amitraz as the active ingredient, but we're starting to see that product fail to control Varroa mites. And that's like what I mentioned before is my research in with my other hat that I wear. And what we've been finding out through the country is that about well, this actually is an old figure. I need to make this a new figure here. I just realized that about a third of the colonies that we apiaries that we sample have Amitraz resistant mites. So you could see there on the right hand side all those colonies, all those apiaries in red. That represents apiaries where the Varroa population has high enough levels of resistance where Amitraz doesn't work anymore. And that's actually crept up to about 40% of colonies, apiaries that we've been sampling. So this is an increasing problem that we have to deal with. Well, nature actually has a way for bees to deal with varroa mites. Natural selection is still operating on bees to this day, and there are populations of quote unquote wild bees that have developed varroa resistance or tolerance, depending on the population. Famously, here in the United States, Tom Seeley worked on the bees of the Arnott Forest near Ithaca, New York, where they were able to find populations of bees that can withstand varroa mites. Similarly, in the Yucatan in Mexico, they were able to find something similar as well, but just in a different kind of bee, mostly Africanized bees, they tend to do well with Varroa. And then another well-studied population are the Gotland bees up in northern Europe here. They exist on this island where they have low Varroa infestation, just based on just without any human intervention. And then finally, the one last one I want to talk about here is in eastern Russia, the Primorsky region. This population here is probably the honeybee population that has the longest exposure to varroa mites. And so we took bees from this region of Russia and bred them here in the United States, and they are the Russian honeybees. So you can see here, there's many different populations out there that can withstand varroa mites without any miticides or other kinds of human intervention. Nature finds a way. Well, when we look at how these populations of bees find low varroa mites, is that there's a lot of different ways to keep varroa levels low. So when we look at the behaviors, there's a lot of them. So one of the things that we're going to talk about tonight, especially, is this infested brood removal. You can see that here in the upper left-hand picture. The bees uncapping and removing that pupa that's infested with the varroa mite. We'll talk a lot more detail about that tonight. Similarly, suppression of mite reproduction. This is a trait where the mites just simply don't reproduce. So you can see here in the upper right-hand side, this is what a reproductive mite looks like. In populations with this suppression of mite reproduction, you have the mite in there, but no offspring. So we'll talk a lot more about that tonight too. Social apoptosis. This one's interesting because this is basically the larvae the pupae self-destruct when they're infested with varroa mites. They self-destruct. And this is something that Kate Eiley from our lab has found in the Russian bees. What's interesting is that larvae self-destruct in Apis cerana, which is the natural host of varroa mites. So this is kind of a convergent evolution between Apis cerana and Apis mellifera in a way to reduce varroa infestation. So I think that's really interesting. In Europe, they have this quote unquote brood effect where there's something in that brood that suppresses the mite's reproduction or it kills the varroa mites. They have no idea what it is, but they found independent of worker bees that the brood has something in it that kills varroa mites. Grooming behaviors, you see on the lower left-hand picture, is a way for bees to knock varroa off of them. They could either groom themselves, like you see here, or they could groom each other. That's a good way for to remove varroa from adults. In that removal process, they could also damage the varroa mites. You can see here on the right-hand side, lower right, a varroa with its legs chewed off. This is a trait seen in the Purdue ankle biter population. And then finally, the last two things that we have here are small colony size and swarming. This basically just reduces the reproductive opportunities for varroa mites because varroa needs brood to reproduce. By having small colony sizes and frequent swarming, it maintains low brood populations, therefore low varroa. However, those are probably two traits you don't want in your bees, right? We want big colonies that don't swarm. But in natural selection, that's a valid way for bees to resist varroa mites. And so this idea for selecting bees with low Varroa was started at the lab in, as you can see here, in 1999. John Harbaugh and Jeff Harris collaborated to isolate and identify the genetics that allows honeybees to survive Varroa infestation. So what they did is they set up a whole bunch of colonies with sister queens that were derived from colonies with low Varroa, and they looked at a number of different behaviors that we'll talk about later. The three most important characteristics that had the highest heritability, I'll mention why heritability is important later, are non-reproductive mites, infested brood removal, and the proportion of mites in the brood. The reason why they want to look at what are the most heritable characteristics is because that makes the breeding process much easier. If there is a high heritability to these traits and behaviors, the easier it is to make progress in your breeding. Low heritability, you're going to be pulling that program for a long time. So in this example, they were able to look at a number of things. So I'm going to walk you through this chart here. So on the left-hand side, we have the characteristic that was measured. And on the right, we have this number called H squared. This is just simply the heritability score. Heritability, or H squared, runs from 0, which means there's absolutely no genetic component to that observation, to 1 or more, which means there is a very, very extremely high genetic component. So the higher the H squared valued, the better it is to breed from, to use that as a breeding tool. So just up at the top here, let's look at suppression of mite reproduction. It was measured 3 times in this experiment. And when they evaluate its heritability, you can see 0.38, 0.06, not great, but 0.46. Anything over 0.25 is what you want to breed from. So those three characteristics, so the suppression of mite reproduction has a high heritability score. Hygienic behavior, very high heritability scores, 0.65. That's going to make the breeding for resistance by measuring hygienic behavior make progress, a lot of progress. This broken legs or bodies, you could see under 0.25. So it's not something that's highly heritable, so we're probably not going to breed from that. But if we look at the proportion of mites in the brood and the mites per 100 cells of brood, this is also two things that we want to look at. And this is important because there are more mites in the brood than there are on the adults. So here's what I mean by that. So if we look at two different factors of Varroa infestation in your colonies. On the x-axis here on the bottom, we have adult Varroa infestation. So this is when you do your mite washes, you put your bees in some soapy water, you shake them around, and you find out what proportion those adults are infested with Varroa, right? Or mites per 100 bees, we call this, or percent adult Varroa infestation. Whereas on the y-axis here, we have percent of brood cells infested. So this is literally taking a frame and opening up 200 cells and finding out what proportion of those cells are infested with a Varroa mite. Well, you can see here on the left-hand side, where the adult Varroa infestation is 0, we could find 5% of the brood that can be infested with Varroa. So the mite washes are a good start, but I think if you want to know the true infestation in your colony, you have to check the brood. So that's why when we're doing our breeding, we're inspecting brood for Varroa infestation, because that is the truest, most accurate measurement of Varroa in a colony. So when they looked at all these characteristics, this hygienic behavior and suppression of mite reproduction, they put it all together in this trait called Varroa sensitive hygiene, or VSH for short. And with VSH, bees can remove infested pupae. On this figure here, when you go from left to right, you see that the honeybee egg hatches and it grows into a larva. And right before that larva pupates, the Varroa foundress infests the cell. The Varroa foundress is in the cell, and when she starts to reproduce, typically at this white-eyed pupa stage, you can see that here in the middle, the honeybees can detect that infestation and reproductive mite, uncap that cell, and remove it. You can see here on the right, image on the right, that worker bee is removing an infested pupa. So it's the stereotyped behavior of detection, uncapping, and removing. And this is what it looks like. So this is the uncapping phase. You can see the worker bee here is starting to chew that cell capping off. And when she gets out of the way, you'll see the hole that she's creating. So that's uncapping. And now here's removal. You can see these workers actively removing that pupa from the cell, cannibalizing her and interrupting the Varroa's reproduction. And you can see here, look at the top here. Look at that Varroa mite coming out of the cell. The Varroa is not reproducing. It has to leave that cell to go find another another pupa to infest. And now we got some grooming. So that's what this looks like. It's detecting, uncapping, and removing. And this Varroa-sensitive hygiene behavior is a subset of general hygienic behavior. All bees with VSH have high levels of general hygiene, but not all bees with high levels of hygienic behavior have VSH. That was a distinction that Bob Denka and Marla Spivak determined in the early 2000s. And so that's one thing I feel we have to consider. The next thing we have to consider here too is Varroa reproduction, this suppression of mite reproduction. So under normal circumstances, like I said, the Varroa will infest that larva right before it pupates and then starts laying eggs. They hatch and develop. As that pupae grows older, you can see here the Varroa in that cell keeps increasing until that bee emerges as an adult and the Varroa offspring also leave that cell with her. Now, it's really important to note here that this has to happen in a coordinated effort in which the Varroa has to enter that cell at a specific time, and then her eggs have to grow and develop so that way they are mated and fertile when they are leaving the cell. And under most circumstances, this is what a Varroa family looks like. We have an egg over here. Now, this is actually a cast skin, but you can see here the foundress Varroa here on the upper left at about 10 o'clock. And then these white patches here are her daughters. There are multiple infestations here. So you can see here a big, huge reproductive family. So this is a reproductive cell. So let's look at the players here. So the foundress, this is what she looks like. And when you do your washes from adult bees, this is what you're going to find. These are the foundresses. These are the ones that invade the larva cell right before it pupates. And they are going to lay their eggs in that cell. They are this characteristic reddish brown color that we're mostly used to seeing. Well, you can tell the different ages of her offspring based on how dark they are. So from left to right, we have very young female offspring, all the way to the right where we have mature, sexually mature female daughters. So you can see here, when they're young, they're almost clear and see-through. And this is all happening inside that sealed pupa cell. You might not ever see these unless you're opening up pupa cells. And then as they get older, they get progressively darker. Okay, so color here is important for aging female Varroa. And just a small little distinction here is that the foundress mite is typically darker than her offspring. You can see here on the right, that is the offspring, which is younger than the mature female on the left. So just that little bit difference in color lets you know if that's a newly emerged female or an older foundress. And that's kind of important because if you have a lot of these soft, pale looking ones, you know that there's a lot of reproduction happening in your colony. With male mites, I don't think any of you have seen a male Varroa mite. It's kind of like a baby pigeon kind of thing. Well, because male Varroa mites only exist in the sealed pupa cells. On the left-hand side here, we have the immature male, and on the right, we have the sexually mature adult male here on the right. And again, you could use color here to age the Varroa. Young males are typically clear and light-colored, whereas sexually mature adult males are this little tiny bit darker brown kind of thing. Males don't leave the cells. Their only job is to mate with their sisters and die. That's why you've probably never seen them. All right, so it's actually pretty easy to tell the difference between an immature female and an immature male Varroa mite. So on the left-hand side, we have the immature female. And this takes some practice to get used to it. But if you see that the body is a little bit wider than it is long. You can see that here. And they have short legs relative to its body size. That's a female. And the males have a more rounded body. It's almost as wide as it is long. You can kind of see that there. And their legs are a little bit, especially this hind leg, is a little bit longer, a little bit more spindly than the female leg. Trust me, it sounds really tough, but once you see 1,000 of these things, it gets pretty easy to sex immature Varroa mites. And this is really important because we need to know if these are present in the cells to measure sexual reproduction in Varroa. Now, I mentioned before that Varroa reproduction has to have been synchronized with the honeybee pupa development. So on the bottom of this figure here, we have pupa stages and days after capping. So you can see here that as these honeybee pupae get older, they get darker. So we typically start looking at Varroa reproduction in pupae that have these white bodies and purple eyes. You can see on the left. And then we can follow that all the way through these other stages until they're just about to emerge, almost looking like an adult at day 12 on the right. Because I said that this has to happen synchronously, we see the expected Varroa families in those cells corresponding to specific ages of honeybee pupae. So if you open up a cell and you find a purple eye pupae, in order for that Varroa mite to be considered reproductive, it has to have a foundress, like you can see here, and then it has to have an immature male and an immature, at least one immature female. Okay. And this is important to see this age of Varroa mites, both males and females, because as you can see here, they have to grow up and age very quickly in order for the males and females to mate. So in this upper right-hand section, the male and female offspring mate at about day 11 or day 12. So if there's any interruption to this developmental schedule, they lose that mating opportunity and are considered non-reproductive. So we have to have a specific pupa stage has to match up with a specific Varroa family age in order for it to be considered reproductive. So this is just another way to show that figure. So again, if we start on the lower left-hand stage here, we can see the white-eyed body with the purple eyes, pupa. And if it's reproductive, we expect to see a female foundress with an immature male and an immature daughter. And this is just basically what I showed you on that previous photo. Now, let's say instead you open up this pupa, and at day 7, the purple eye, white body stage, we only have a foundress. And we don't see that male offspring occurring until day 10. Well, you see here at day 12, they have a male, a mature male, but the female offspring is sexually immature. They have no mating opportunity. Therefore, we would identify this as a non-reproductive event because that female offspring, this little lighter colored one, is not mature enough to mate. They lose the opportunity and it's non-reproductive. You can see here, it has to happen in the Varroa reproduction has to happen synchronously with the pupae age in order to be considered reproductive or not. Okay, so here's your quiz for tonight. We're going to go through this. I'm going to show you some examples of a pupa stage with the Varroa family that we find in that cell. And just to yourself, you can tell me if it's reproductive or not. So in this example here, we have a white-eyed pupa, white body pupa with purple eyes. And in that cell, we find a foundress Varroa with an immature male and an immature female. Is this reproductive or not? This would be a reproductive event. So if we open up that pupa and find that Varroa family, we would say that that Varroa is reproductive. Well, let's say we open up that pupa cell, same age, white body, purple eye, but we only find the foundress. Is this a reproductive mite? Yes or no? This would be a no. Because remember, it has to have that male and female offspring at this stage in order to allow them enough time to sexually mature and mate. Okay. Now we have a pretty old pupa here. It's starting to look like an adult. You see here it has the black body and tan abdomen. And then we go in there and we only find two foundresses. Is this a reproductive mite? Yes or no? If you said no, you're correct. Because at this stage, even though we have two female mites in this instance, this is an instance of a double foundress infestation. Both of them are non-reproductive. At this stage, we would expect to see sexually mature males and sexually mature females offspring in order to mate. The absence of them tells us that these Varroa are not reproductive. Okay, here's a good one. So we have a pre-emerging pupa, looks like an adult pretty much. And in that cell, we have a foundress and two female offsprings. Is this reproductive? Yes or no? I'm going to say yes. So in this example here, we do not have a male. Remember, I said males only exist in the cells to mate with their sisters. The absence of the male, since we can't confirm it, we have to assume the male is there because the male egg is the first egg that's laid. Because sometimes the males are hard to see. They might be on the pupa. They might be hiding in the mess at the bottom of the stage. We're going to be conservative in this case and call this yes. For breeding purposes, it's better to say it's reproductive than to say it's not reproductive. You want to err on the side of caution here. So this was a tricky one. So even though we don't see the male, we're still going to call that reproductive because those females are the proper age. All right, so let's open up a cell where we have a little bit darker pupa. We got some black eyes here. And then we see the foundress with the male and female offspring, but they're immature. Is this reproductive? Yes or no? Yes, that is reproductive. That is the Varroa family we expect to see at that age of pupa. So that is a reproductive mite. All right. Well, here we go with a pre-emerging pupa. We have a foundress and a male. All right, I see Brian shaking his head no, because yes, it laid a male egg and hatched, but it's way too late for that male to mate with his sisters. They're not even there. So that's a non-reproductive mite. Okay, so pretty young pupa, we have two foundresses, two males, two immature female offspring. Reproductive, yes or no? Yes, that's definitely a reproductive mite. Well, both of them are reproductive, actually. Typically, you'll have one male offspring and two or three female offspring as well. So this is example, this is what we expect to see in the event of a double infestation. You can't see that, especially late in the year. Okay, then finally, let's say you open up, you find a white body, purple eye pupa, reproductive, yes or no? This is what you're going to see a lot of. This would be no reproduction because there is no mite. You can't have non-reproductive mites if there are no mites. So this is what we look at for when we go to look at these cells to find Varroa in terms of infestation. But then if we find Varroa, they have to be non-reproductive. So this is pretty complicated. This is why this breeding is pretty tough. Well, we use those pieces of information, the number of Varroa in the cells or the proportion that are non-reproductive, to base our breeding decisions. So on the left-hand side here, let's say we open up cells and we find that they're infested with Varroa mites. However, if a high proportion of those Varroa mites are non-reproductive, that's actually good for breeding. So on the left-hand side here, hypothetically, let's say we found 10 mites and all of them were non-reproductive. That would fall over here to a colony score of 4. That's the highest score we can assign that colony. Because even though it has mites, they're all non-reproductive. that's a good breeder. So that's why I say definitely as a breeding source. Well, let's say that you went in and you're only able to find reproductive mites. So if you find 0 mites and 200 cells, well, that's still very low of Varroa infestation, and that's the most accurate measure of Varroa infestation. Even though we can't measure non-reproduction because we don't have any Varroa, that's the name of the game right there. We don't want to find any mites. So if we find 0 mites and 200 cells, we're breeding from that. That's a gold star, no doubt about it. These scores are pretty stable too. So all you do is need to measure colony once, and they'll get the score. Well, then let's look at the next tier. Let's say 50 to 79% of those Varroa are non-reproductive, or you find 1 mite in 200 cells. It could be either or, because, and I want to make a distinction here. You can find colonies that have lots of Varroa, but as long as they're non-reproductive. But if you find reproductive mites, you want to find very few of them. So in this situation, the second tier, we would be breed from that, because that might be the best you find, right? You might not always find these high tier 4 breeders. But yeah, we'll keep that one. Well, then finally, what we might want to consider a breeder is something with 30 to 49% non-reproduction, or 2 to 4 mites in 200 cells. We're going to put that one in the maybe pile. Again, that might be the best one that you have in your operation. So it's better than nothing. So that goes in the maybe pile. Anything below that, I really wouldn't breed from. It's not worth your time. Usually when we're opening up cells and we find 5 mites, we just stop because we just know that that's not going to be a breeder. So we have to either find non-reproductive or just very few mites. That's the name of the game. That's how we base our breeding decisions. So this sounds great. Okay, we can have bees that have low Varroa mites. Well, let's look at how they perform as colonies. So when John and Jeff started this program a long time ago, they were able to isolate this VSH trait in a very, very specific small population. It was very purebred. While that's good, there are some colony performance issues. Well, when we look at Varroa mites, the resistant ones have 44 times lower Varroa than the control colonies, the unselected bees. So that's really good. 44 times Varroa, lower Varroa, sign me up. Number of Varroa in the cells, so this is opening up cells, 70 times lower Varroa in the pupa cells. That sounds really exciting. However, there are trade-offs. The resistant bees had 35% less capped brood area, so that's quite significant. And then also too, they are 44% lighter. They didn't raise enough pupa to adults. They didn't harvest enough honey. So you can see here, 44% lower colony weight in these resistant honeybees, this original VSH stock that they made. And this is kind of expected with breeding, because selective breeding is, by definition, reducing genetic diversity. First thing you want to do is eliminate anything that's unnecessary and isolate that trait. But in doing so, there are often consequences. And it's not something that we see with this VSH honeybee stock, but this is agriculture, right? This is something I mentioned here. They have typically these spindly little colonies. There's a compromise. Well, we see this with bananas. Bananas are clonal. They're mostly one variety. While they produce lots of bananas that keep our shelves filled, they are susceptible to this fungal disease you can see here. Similarly with chickens that are bred for meat production, they focus mostly on white meat production. So that part of their body grows larger, but the rest of its body doesn't keep up, leading to these prone positions in certain members of that flock, which can be pretty bad for them. So this is something that's good for making lots of white meat chicken, but maybe not the best thing for the chickens themselves. And if anybody has any purebred pets, you know that there are a lot of issues that come with purebred, because again, you are by definition reducing genetic diversity. And especially like with German Shepherds, this sort of posture, while this is something that breeders wanted, it's actually really bad for those German Shepherds. It causes issues with their hips. And you name any other purebred breed of dogs, there's always something wrong with them. So this is, again, something expected and just the natural outcome of selective breeding. Well, what you want to do as any reputable breeder of anything, whether it's plants or animals, you want to improve that stock. Now you have something that is desirable. So what they did here at the lab, this is again going back 20 something years, is that we were able to work with beekeepers to take this really inbred, highly resistant stock, and they were able to work with beekeepers to incorporate traits that they like. So what we did when I say we at the lab, I mean John Harbaugh, Bob Danka, Garrett Dodds, and Jeff Harris did all this work, and I'm just telling you about what they did. They took this stock and they worked with other beekeepers and they crossed it. So those queens mated with their drones to create this outcrossed population. Those queens that were mated with their drones were then brought back to our lab and then back crossed to that resistant stock. And then those daughters, this F2 population, this back cross population, was evaluated for low Varroa and large bee populations. And then the following year, those queens were sent back out to the beekeepers to be crossed with their drones. Her daughters were sent to beekeepers to be outcrossed with their drones and then brought back and forth to the lab. This process was repeated for 3 years. And in doing so, we're able to bring in all that good stuff from the commercial beekeepers, what they liked, but have that VSH trait in the background. And that final product was this stock we call Pol-Line. Pol-Line has high levels of VSH behavior, but it also has rapid population growth because with commercial beekeepers, they want big colonies for almond pollination, with really high honey production, high overwintering survivorship, and gentleness. These bees are so gentle, you can work them sometimes without a smoker and just a veil. It's really nice to have something that's this gentle. So these are things that are really good improvements on the original resistant stock. But however, at the lab, this population has been maintained with rigorous selection and propagated by instrumental insemination. So this is something that we know works at the lab, but in small numbers in a very limited geographic range. So we want to improve on this. So when we're looking at things that we can do for Pol-Line and how they stack up against other stocks. So some of the advantages of Pol-Line include high overwinter survival. I'll show you the data. Lower virus levels. We'll talk about why they have lower virus levels. Hint, hint, it's Varroa. Reduced miticide use because these bees have VSH. You don't need to apply the miticides as much. And if you got a miticide bill last year, I'm sure you know what I'm talking about. And so as scientists, though, it reduces the impact of Varroa viruses and miticide on experimental design. So that makes our experiments a lot cleaner. So there's some definite advantages. Some things that are comparable is that these Pol-Lines are basically Italians. So if you know Italians and like Italians and that's what you like to keep, then this is your bee, this is your stock. They're not Carniolans, they're definitely Italians. So they're Italian in terms of management. They have comparable colony sizes. I'll show you data about frames of bees. They have good brood buildup, so they're always ready to go, especially for almonds. They have comparable honey production. They don't make more, they don't make less. They make about the same. However, again, there are consequences, there are trade-offs. So some limitations of this stock is that they have high Varroa sensitivity. The typical thresholds for treatments with Pol-Line, it's way too high. I'll show you what I mean by high Varroa sensitivity. Very few Varroa. They can totally tolerate very few Varroa. There's limited availability. You can't just call up somebody and say, hey, I want some VSH bees. We're working on that. So we want this to be everywhere so you can get these VSH queens from anybody at any time. There's rapid genetic dilution. So when these VSH queens, their daughters outcross to non-VSH drones, those F1 queens are okay, but the second round that her daughters then outcross again to non-VSH drones, this trait is gone. So just 2 generations, this trait could be completely gone from your population. Wrong way, wrong way. So we wanted to find out how this actually works in the field. We didn't want to make sure this wasn't a laboratory parlor trick. So this was one of those big studies I told you about. So this was started in 2017. Bob, Mike, and I from the Honeybee Lab collaborated with Judy from the Beltsville Lab and Kristen and Dan from LSU, and we got a grant to do this research, and it's been published already. This was looking at minimizing the impact of Varroa mites and mite-borne pathogens on managed honeybees. So what we did for this project, and this is 2017 and 18, we did this huge project with the migratory commercial beekeeper, and we were able to use 171 colonies of Pol-Line and 191 of the unselected commercial stocks. So huge numbers of colonies, and this is what 191 putting queens in 191 colonies looks like. So you can see that here. We then took those colonies and moved them around the country. Well, not we, the beekeeper that we collaborated with. So we started these colonies in April of 2017. We then measured a bunch of things about the colony performance in South Dakota and North Dakota in June of 2017. We then went back up to the Dakotas in September, right after that honey was pulled off of it. And then we looked at the bees where they overwintered in California or Los Angeles in December. And then in February, we went to California right when they were in the almond orchards, or right up the road with the ones that were near our lab. And then the final observations for this experiment on an annual basis occurred in April of 2018. So this is the full beekeeping circuit from colony birth to hopefully survivorship or death. So we're trying to find out what's going on at these 6 time points throughout that beekeeping year. And when I said we measured everything, I mean we measured everything. Today, I'm going to tell you about the data for the measurements of Varroa infestation and viruses. Those 2 things are usually linked. We also looked at queen status, bee population, so frames of bees, honey production, brood area, pollen and nectar stores, pesticide exposure, total lipids, oxidative stress, gut microbiome, gene expression of very important genes. Basically, if there was something to measure, we measured it. So this was one of those $2 million projects that we worked on because we were doing so much sampling and so much data analysis. Well, let's just look at what the major findings from this study were. So on the x-axis here, we have month. So this is May of 2017 through April of 2018. And on the left-hand side here, we have percent colony survival. So in May, 100% of our colonies survived, which is fantastic at the start of the experiment. And as you can see here, through June and September, Pol-Line and commercial had very similar survivorship. But then after September, you can see here the commercial colonies declined very rapidly. Whereas at the end of the experiment, only about 20% of the commercial stock were surviving. Whereas with Pol-Line, about 60% of the colonies survived. So we had 3 times higher survivorship in the Pol-Line colonies compared to the unselected commercial stock. Now, these colonies were all treated the same. The only thing different here is genetics. Okay? Well, what are the consequences of those genetics? Why did Pol-Line survive at a higher rate? Pol-Line survived higher because they had fewer Varroa after honey harvest in September. On the x-axis, we have Pol-Line, our resistant stock, then the unselected commercial stock. On the y-axis here, we have Varroa infestation, measured in mites per 100 bees, or you could just think about this as percent infested. And you can see Pol-Line only had about 2.5% infestation across all the colonies, whereas the commercial stock had over 8 mites per 100 bees in September. So about 4 times less Varroa in Pol-Line. Well, there were some stock differences in Varroa sensitivity. So again, we have Pol-Line on the left-hand side, commercial on the right, and Varroa infestation on the y-axis. The white bar here under Pol-Line indicates the Varroa infestation that in September that was associated with those colonies surviving. So if a Pol-Line colony had less than 2 Varroa, that colony survived to the end of the study. Whereas if they had 4 mites per 100 bees, as you can see here by the red bar, those colonies from Pol-Line were dead by the end of the study. However, the commercial bees, they actually survived this study with 4 mice per 100 bees. So the same amount of infestation kills Pol-Line, but the commercial bees tolerate. It took a lot more Varroa to kill the commercial colonies. You can see here about 10% infestation was needed in order, on average, for commercial colonies to die at the end of the experiment. So yes, stock does matter. The Pol-Lines can keep Varroa levels low. They got to keep them even lower than the commercial stock. So this is something that I didn't expect to see to this significant degree. Stock does matter. Well, we know that these colony losses are mostly due to Varroa. So if we look at this figure here, the Pol-Line and commercial colonies here in the black bars, and this is total colony losses by the end of the experiment. So in Pol-Line, we lost 54 colonies, or about 30% of our losses, whereas in the commercial, we lost about 65%. Well, if we look at the proportion of colonies that were lost with high Varroa, so if we go in Pol-Line and we look at all the ones that had 4 mites per 100 bees or more, and the commercial stock, we look at the ones that died with 10 mites per 100 bees or more, you can see here a significant majority of the colonies that were lost within each stock was due to high Varroa. So this is why at the beginning of this study, I said about 70% of our colony losses are due to Varroa, and this is the data that gets us to that. So most colonies are dying because they have high Varroa mites. Very few of them survive with high Varroa. Well, when we looked at the viruses, well, we were thinking, well, maybe the viruses have something to do here. Well, if you look here, is that on the x-axis here, we have chronic bee paralysis virus, deformed wing virus A, deformed wing virus B, and then we have the log mean viral quantity. What that means is that every number here is actually a 10-fold increase. So between 6 to 8 on this scale, since it's a logarithmic scale, it's actually 100-fold increase. So it's on a log scale. As you can see here, Pol-Line had lower levels of chronic bee paralysis virus compared to the commercial stock. Similarly, lower levels of deforming virus A and B. And this is due to the fact that these viruses are transmitted by Varroa. The idea here is that lower Varroa means lower viruses, and that's exactly what we see in the data. So on this figure here, we have the September Varroa infestation on the x-axis, so low Varroa, high Varroa. And then we have the amount of deformed wing virus in those bees. As you can see here, as your Varroa population increases, so does the concentration of deformed wing virus. So the deformed wing virus is almost exclusively transmitted by Varroa. So if you keep Varroa levels under control, you'll have less deformed wing virus. So this is another benefit of having lower Varroa. Lower Varroa usually means lower viruses. Not all of them, but the ones that are major players, chronic bee and deformed wing. All right, so let's look at some of the things that are comparable. Adult bee population. So this is frames of bees. So how many of those frames are filled with bees? And as you can see here, at no time point was there any significant difference in the number of frames of bees between commercial bees, shown in the black bars, and Pol-Line in the gray bars. There are some numerical differences, but they're not statistically significant. So this shows us that these are really good for commercial beekeepers, especially for almond pollination. They produce comparable amounts of honey. So commercial and Pol-Line, those two bars, the honey production in terms of pounds per colony, no significant statistical difference from them. So when in the same environment, they're going to produce the same amount of honey. So some of the take-homes I wanted to have from this study is that high Varroa infestation is the major cause of colony losses. This is something that we saw in this study, but every other study that we look at, we have to look at Varroa. It's the major reason why colonies are dying. Yes, pesticides, other parasites, land use, all that stuff is very important. However, we can explain 70% of the reason why they die simply from high Varroa. Bees selected for VSH behavior have reduced colony losses under migratory beekeeping conditions. This shows that it's not something that we just do here in Baton Rouge, but this is something that is ready for prime time in commercial beekeepers. And I'll talk to you a little bit more about what our plans are for rolling that out. There are stock differences in sensitivity to Varroa infestation. I was not expecting to see this, but what this tells me is that there is no safe levels of Varroa unless you can identify how many Varroa it takes to kill your bees. So to me, I think less is best when it comes to Varroa infestation. There's low levels of Varroa transmitted viruses due to lower Varroa infestation. Um, and I didn't have a chance to talk about it, but we tested if Pol-Line somehow has virus resistance. Nope, they do not. They have lower Varroa. They have lower viruses because they have lower Varroa. And then they're comparable to commercial beekeepers, commercial stocks in almost every other phrase in terms of bee populations and honey production, things like that. So this is good. So then this is like at this point in this talk, I usually feel like I'm doing an infomercial because people say, well, where can I get these Varroa resistant honeybees? There has to be a better way. Well, there hopefully will be sometime soon. I'm gonna skip over that. And that's kind of what the goal of this project is that we're working on for at least the next year. So this is a USDA area-wide grant that we got to improve honeybee health and food security through breeding and IPM-controlled parasitic mites. This was a grant awarded to Mike Simone Finstrom, Ariane Avalos, me, and Liz Walsh at the lab. And then we are collaborating with all these other people across the country to do a major improvement of honeybee breeding. So with this grant, we have 4 major goals. The first is to train and perform people to do VSH evaluations and selective breeding. So that way you could raise these bees and develop your own stocks. And then also to the commercial queen producers will be incorporating this into their breeding programs. Second is to develop better genomics tools for selective breeding. So that way we don't have to wait a whole year to do one generation. We could do 2, 3, 4, 5, 6 generations in a year if we could just look at the DNA. So Ariane is in charge of that. Improve detection of amitraz resistance in Varroa and develop practical treatment regimes and IPM practices. This is my major contribution to this project. And then Liz is looking at the economics of using Varroa resistant stocks because beekeeping is at the end of the day, it's still a business. So we have to find out if it makes economic sense. And spoiler alert, it's really, really economically beneficial. So this is what we started doing this project last year. It was going to run to 2028, but there's been our funds probably aren't going to get renewed, and that was known from the start. This isn't anything new. So this was just it's probably not going to be renewed by Congress. So it's not a new cut. We measured Varroa infestation and reproduction in the brood from top 50 performing colonies in a beekeeper's yards. And then we went and looked at the Varroa levels in those colonies. Well, when we looked at some of these large-scale commercial queen producers, I would have had an expectation that we might find, 10, maybe 15% of their colonies making grade as breeders. Well, we actually found about 30% or more actually made grade as breeders. So these are just 4 anonymous beekeepers, and then this is their percent of those colonies that made that we would breed from. I was expecting 10 to 15%. You can see here by this band, but what we actually found was about 30%. So we know this trait is out there. We just have to identify it and encourage selection from there. So we're going to work with these people again this year, and hopefully this number will be about over 50% this year. We want this to keep going up and up and up, and it will if we just identify colonies that have VSH and breed from it. So this is exciting. And we know it can be successful because, like I mentioned before, Marla Spivak from University of Minnesota had tremendous success at getting people to breed for hygienic bees. So the beauty here is that this kind of sets the stage for selecting for VSH because VSH bees need to have this hygienic behavior. So the stage is set for high levels of success with this program, unknowingly by Marla 20-something years ago. So yes, we know this can be successful because her program has shown that it can. Well, that's what I wanted to finish up my talk here about tonight. And of course, I didn't do all this research by myself. I had a lot of great people that work with at the USDA, but then also at LSU. They are major collaborators. But none of this could have been done without our industry partners. You see some of those guys on the lower right-hand side. I mean, it's a pleasure working with these people. They make fun of me because this is something they do every day, and I'm out there trying to help out. I'm just getting in their way, and they're yelling at me and stuff. But it's really fun to work with these people and do something that ultimately benefits them. So with that, um, I'll finish up, and we'll go to the question session section.
All right, thank you so much, Dr. Rinkevich. I actually, just for a little bit of background knowledge about me and what I'm bringing to this conversation, I'm in the Master Beekeeping program here with Colorado State Beekeeping Association, and my master project is around Varroa sensitive hygiene and specifically the UBO assay. So I've learned all about the Harbo assay and freeze kill assay, and I've been trying the UBO assay. Will you talk a little bit about the time frame that's required in providing your colonies with a treatment-free period before getting a valid data point on the Harbo assay? And then as you go on, will you talk about your hopes and thoughts about the UBO assay and how that works into kind of different levels in the industry, like the sideliner, backyard beekeeper, commercial, how they might utilize these resources? That's a lot, sorry.
Oh no, this is great because I have all the answers right here already. Because trust me,
Cool.
you're not the first person to ask these questions. Um, so the best way to do this is we think that 8 weeks post your last treatment is adequate enough time to successfully evaluate Varroa infestation in your colony. And the reason why we know that is because when I showed you those, let me just go back to it. When I showed these with these beekeepers, we were getting higher than I thought. And I said, well, maybe it was too close to our last treatment, and it was 8 weeks since our last treatment went on. I thought, well, maybe the treatment was really good and it kept Varroa levels low. That's why we're seeing this. Well, what I'm not showing here are the other colonies that had like 20% Varroa infestation in that same time period. So what that tells me is that 8 weeks is enough for those Varroa populations to build up to where they need to be. And I think this signal was true. That being said, though, 8 weeks seems to be sufficient. Longer the better. When you're doing this breeding program, you really want to eliminate that as a potential variable. If you could take that leap of faith and go treatment free in order to evaluate your colonies the most accurately, I think that's setting yourself up for success.
So
Now,
just to clarify there, to get the most valid Harbo score, you would even remove mechanical treatments like removal and even like spring splits. Those would have an impact on your scores.
it can, I it can, I can. However, we know that we could still get valid data because what we did here, and the way that we know that this is that 8 weeks from that last treatment is adequate, because when we did these scores here in this was in August, we looked at those same, well, not we, some collaborators went back and I said, hey, could you go and check those colonies to see if that was really a zero? And when it was a zero in August, was still a zero in December and in February. So that's why I said these scores tend to be very stable. And
Yeah.
also too, in that time frame, they were not treating, because I told the beekeepers, I said, okay, we have potential breeders here, but in order to confirm, to make sure that these are going to be really good, don't treat these colonies. So they went from August, well, they went from last ones went on in April. May. The last treatment went on in May. So they went from June, July, August, September, October, November. So 8 months. Well, seven. I can't count. 5, 6,
Yeah.
7 months without
Oh,
treatment. So yeah,
okay.
yeah. The longer you can go
Thank you.
without treatment, the better in order to get that score. But I think the data clearly shows that 8 weeks is suit adequate.
Thank you.
And then the second
And
part
then
was the
yeah,
breeding with
the
the
UBO assay. Yeah.
Yeah, UBO, that's being developed by Kara Wagner. And there are
Hmm.
uh, some research that's coming out about how that tool can be used. And Kara will tell you this herself, is that there is a seasonal component to it. So typically the best UBO scores are going to come when there is a good nectar flow and the bees are happy. So usually for us, I don't know what it's like in Colorado right now, but we're flowing like crazy. So right now is the best time to do your UBO test. She said in the summertime, when there's a dearth, and even in the fall, if there's any kind of dearth, the bees aren't going to respond like they normally should. So there's a seasonal component. Second thing is she had a dosing issue with her assay that it would sometimes either over apply or under apply. Well, now she has that worked out where every single and if you've used it before, never had to hold the gun up a little bit, and sometimes it would
Yep.
spray perfect, and other times. So she's refined that, and
Yeah.
that's got a lot better. So more consistent responses. So
She's
she's
also
always
done a really nice. Sorry, go ahead.
so she's just improved that test so much where it's a lot more consistent. And I think that a
Yeah.
more consistent application and understanding the nuances of it are going to really let you interpret that data better.
And that's kind of what I was going to say. She listened to the response of people who used it really well and listened to their struggles that first year it was out and made some really nice adaptations to it. So that was really great to see. It's also hard with a product like that to know when to take it to the public, to know when it's ready, knowing that there still might be some improvements to make on it, but knowing you want to release this tool to beekeepers to for them to be able to use it, knowing that it might not be perfected yet, but good enough to be able to get some good outcomes for people.
Well, it's just like any new technology is it's almost never perfect on the first rollout, but we'll figure
Yeah.
it out and we'll learn how to use it. And the reason why I want us to try to learn how to use it is because it would give us another way to evaluate our colonies earlier in the year. Because in order when we're doing harbo tests, these cell inspections, we got to wait till August or September to do this. I'd rather be doing that right now.
Do you ever do Harbo tests of colonies coming out of the winter in the spring?
Not normally, because
Okay.
typically Varroa levels are low at that time of year. The reason why we do it in
Yeah.
September, August and September, is because that's when Varroa levels are their highest, right?
Yep.
Usually coming out, there's not, there's, the Varroa go in these boom bust populations, and usually coming out of winter, they just have low Varroa on their own.
Gotcha. All right. I don't know if this is a loaded question or not. It used to be called SMR, and then it was changed to VSH. Some people still kind of distinguish between the two different behaviors. Some people just say it was a change in name. Do you have a perspective on that?
Let me guess, you've been talking to Corey Stevens.
Maybe.
Yeah, no, that's true. So what we're not in all fairness, we are not really measuring VSH in this, truth be told, because if you actually wanted to sit there and watch the frames and watch them actually opening up cells and removing pupae,
Yeah,
we've done it. We're never doing that again. So
yeah.
what we're measuring is like a surrogate or like a kind of what's I looking for a, a yeah, a surrogate measure of that VSH. So VSH specifically is referring to them removing the pupae. However, just uncapping the cell is enough to interrupt mite reproduction. So, and we can measure SMR as part of this Varroa non-reproduction kind of thing. I think we need to come up with a better name for it that's inclusive of both VSH and SMR because that's
Yeah.
really what we're measuring. So maybe this is um, we call this Varroa resistance score or something. So yeah, it's
So I'm
tricky.
glad you brought up uncapping and capping as part of that conversation. You consider the removal of pupa and uncapping and capping as different hygienic behaviors with the same outcome. Well, I get, would it be the same outcome? No, the outcome would be the pupa stays alive just in the uncapping, recapping. Is that correct?
Yes, but if the but with yeah with uncapping and uncapping the pupa can survive, but uncapping and removal the pupa's out of here. Um, and there's been some populations in Europe that well no we have them here in the United States that they prolifically uncap right,
Yeah,
and that's
yeah.
enough to trigger Varroa non-reproduction. So the removal might not be necessary for this behavior, but what happens is is that the removal is usually because that is a reproducing Varroa. And when it's reproducing, it's also transmitting viruses, and that's activating that hygienic behavior because there's
Okay.
some viruses transmitted with it. Yeah.
And is it correct that the theory is the uncapping changes the humidity level with inside the cell, which impacts the reproduction? Okay,
I don't know.
good answer.
Hopefully, someone does the
Um,
research because trust me, the mechanism of SMR is I don't think anyone has a good answer. Well, we have
yeah,
ideas, but I don't think anyone has data.
gotcha. Now, social apoptosis. You talked about the larva self-destructing.
Mhm.
So could you talk about that more? Describe that? Like, are any of the bacteria, viruses are self-destructing in that process?
No, the what I mean by that is that the pupae larvae or pupae just dies. Um,
Okay,
it doesn't like MacGyver blow up or anything like that. It just
right.
stops living.
Okay.
And when it just dies, the roa just doesn't reproduce. So
Is
yeah.
that cell capped open then and removed?
Nope.
What happens to that cell?
So I'm thinking Kate's paper. Um, I don't think they evaluated that because they were using gel caps to measure if that pupa was alive or not. I don't know. I'll ask Kate
Okay,
tomorrow. That's a good question.
thank you. Um, Now, John Harbaugh on his website has a really good description of the process of the Harbaugh assay that feels like a pretty old document, though. Has there been any evolution in best practices of the Harbaugh assay, or is it pretty much the same as what's described in that paper?
It's one of those things. Ain't broke, don't fix it. Um,
Yeah.
because we've had other people think about improving it or making it better, and I don't know, man. It's hard to beat the old Harbo test. Yes, it takes time. So with us, is me and my technician when we do these tests, name of the game is speed. If you're trying to evaluate 50 colonies, it takes about for us, it takes about 20 minutes a colony because we just do hundreds or thousands of these every year. So we could do a colony in about 20 minutes. Um, and it just that's what it takes. Some people have come up with other methods and we've tried them, and I don't know. So there were some people that were just taking like piece of duct tape, uncapping a bunch of cells and seeing if they're infested. But what happens is
Hmm.
are not evenly distributed through the colony. They're kind of patchy. So if you're sampling from a patch like this, but the row are over here and over here, that kind of is going to be tricky to find them. Because that's what we try to do is when we're opening up cells, we're just like plucking randomly all over it because they are patchily distributed. I don't know. It works. It takes time. Like all good things take time.
So let me just clarify what you just said. When you're doing the Haribo assay, you're looking for a certain age pupa right around the purple-eyed stage, but you're not looking up for a particular cluster. You're trying to find representation across, is it across one frame or multiple frames?
One frame, both sides, as long as
Okay,
it has the right
okay,
age. So it has to be purple eye or later.
gotcha. Thank you.
Yeah.
Um, yeah, let me get to some of the questions
Yeah.
in the chat as well.
Mhm.
Um, Here's one from Deanna. Would you quarantine to help stop the spread of varroa mites? I understand that New Zealand did this and it worked to eradicate varroa mites.
It
Well,
depends on how early you identify the infestation. A lot of times, rows been in there for a lot longer than you even knew. So, for example, here in the United States, we say that I think the first infestation turned up in 88. Well, in order for that to be found, it's probably been here a lot longer. They anticipated, even like in Australia, where they had the infestation, they found it in so many different places that weren't connected from beekeeper trade that they think, okay, this stuff might have been here last year. We just didn't find it or look for it. Um, I don't know. I think there's been a lot of quarantine efforts for Varroa that just don't succeed. So, like in Australia, just, I don't have any specific source for it, but what they're trying to do is quarantine and colony destruction, and they had to destroy so many colonies that they ran out of money really quick, and they just couldn't contain it. So it was just a lot of colonies died for no reason. Um, I don't know. I think that especially with Varroa mites, it's part of beekeeping. There's no way to stop it. We just got to manage it.
You
That's
know, it seemed like looking at it from across the globe, it seemed like an exercise in futility of anyone that's familiar with how Varroa works.
mhm.
But here we are with trophy mites on the horizon. And I'm not sure quarantine is the question I want to ask about trophy mites, but as far as hygienic behavior, do you see any kind of translation from Varroa mites to trophy mites with this kind of VSH re resistant behavior?
So I was in Korea last year to study that exact question, and we're gonna
Cool.
do some stuff next year. We found that colonies with higher levels of VSH or SMR had lower trophy, and this was in their bees that they had over there. So I think we evaluated about 50 colonies. So we found a gradient of that behavior, and sure enough, ones with higher VSH had lower trophy. So what we're doing next year, because this year's got away from us, is we're going to send these Pol-Line queens over to our collaborators in Korea and run that experiment.
Oh,
So if we have, a major tool in the toolbox, maybe trophy won't be as bad.
right. Awesome. All right,
Yeah,
Paul asks,
there's
go ahead.
so much work we're doing on trophy. So when it comes here, we're gonna have a sampling network to make sure we could identify infestations very quickly, rapidly, and cheaply.
Yes. Paul asks, it seems like for the hygiene management to work, it comes down to timing. They pull the pupa. Does your research have any insight as to when the worker bees are most likely to detect and pull the pupa? It seems like they're detecting early enough if hygiene is effective.
Yeah. So the workers are um detect, usually it's white eye or pink eye or purple eyed pupae that they're detecting and removing from. And the idea here is that that's when the Varroa are feeding on the pupae, and it could be two things. There could be an injury signal from the pupa because they leave a pretty gnarly hole in that pupa, and maybe that damage signal is what they're cueing it into. And then there's other research that showed that what they're actually removing is when the viruses start to reproduce in that pupa, that sends off some kind of signal that the pupae gives off that is detected by the workers. So it's either a pupil damage signal or a virus reproduction signal. What the exact molecule that does that is very likely to be the compound that's used in the UBO test. But Fanny Mondet over at basically the French version of our lab showed that it's actually a little bit more complex chemistry than just one chemical and one isomer. So that was a really interesting paper. So it's probably a mix of chemical signals due to damage or virus reproduction at that white eye, pink eye, very early purple eye stage. They typically don't remove older pupae that are infested. Typically, but they can.
Anne asks, did the Pol-Line bees receive the same Varroa management protocol as the other commercial stock bees? I believe they were both untreated throughout that study. Is that correct?
So yeah, so they were managed in the exact same way as the commercial stock to a T. The only difference, though, within those stocks is that some of them we treated with the standard treatment, and others did not get that treatment. And what we found is in the commercial stocks that were treated had lower Varroa, but Pol-Line that was treated did not have significantly lower Varroa. So what that told us is that VSH behavior is your Varroa control program. You don't really need to put miticides on. It's not going to get them that much lower.
Thank you. Deanna asks, has nutrition been a consideration? What did beekeepers overwinter with? Honey, sugar board, some commercial formula. Did Dr. Priya Basu is researching honeybee nutrition. So
Yeah,
I'm
so
not sure if she's referring to that particular study or what.
yeah, so there is some idea here that there is a nutritional component to it because there is some seasonal differences that we definitely see, especially like with Carol will tell you about with the UBO test. But don't have exact numbers on it, and that's something I actually talked to Priya about. This is kind of funny because these are the same things that we talk about all the time, so these are great questions. But I talked to Priya about this. I said, maybe bees that have better access to nutrition will have lower Varroa, and we saw some of that in the data. So in these longitudinal studies, we have different apiaries set up across different gradients of land use, and some of the ones that were near areas with good forage had there was a trend towards low Varroa. It wasn't statistically significant and it was pretty noisy, but there was kind of some suggestion that says that maybe nutrition has something to do with this. Really don't know. No definitive answers yet, but there was enough smoke to say that it deserves more research for sure.
So
Yeah,
does this get into the world of epigenetics where nutritional markers might turn on specific genes that activate particular behaviors? Is that the
yeah,
implication here?
yeah. I mean, and it makes sense, right? But we don't have any data on that, and that's something that I think is a real strong missing component here because I'm a big advocate of natural forage. If you feed your bees, how expensive it gets.
Sure.
Um, but I'm a big advocate of that, so that's why I'm always trying to incorporate that land use into our studies, just to kind of have some kind of potential information on nutrition. So we do quantify pollen and nectar stores in these colonies to see if it's a quantity issue. Um, and do we? I don't recall if we've ever seen any kind of signal based on those two things, but I know the landscape use, depending on potential available forage, they tend to, well, not tend to, there's a suggestion, not definitive by any remote means, but it does warrant further investigation. And mostly because I think it's because when they have adequate nutrition and they are well fed, the colonies are usually dividing their labor in a more effective and discrete manner. So therefore, you're going to have more. The workers are going to be performing certain tasks at those ages. Now, when you have a nutritionally deprived colony, they're increasing their foraging force to compensate for that. So they skip that nurse stage and that worker stage, so therefore VSH is lower. So there's a logical component to it, but as far as hardcore data, peer-reviewed data, I'm not aware. Hopefully, someone will do that study soon. Talk to Priya. Tell,
Thank you.
tell someone, tell Priya to get some money so we can do the study.
We kind of talked about this a little bit before, but Brad asks, do you see this VSH research as a step towards responding to tropolalaps, or is that a bridge to be crossed later? I don't know if you want to add anything to that.
Yeah, no, hopefully we'll be doing that next year. We just started our collaborations with our cooperators over in Korea. I think that's gonna be very interesting to get those data. What's interesting is because with Tropy is that they mostly live in the brood. They don't really hang out on the adults like Varroa does. So we need to have something that targets the brood. Well, what targets the brood? VSH. And like I said, is when we looked at colonies that had hygienic behavior over there, there was a, it was not statistically significant. It was what was what I'm looking for. So in science, we only really we tend to say there's a difference when it's statistically significant. But when that line is going in that direction and maybe one of the points is a little bit off and your p-value is like 0.06, I always just say we need more samples. So that's what
Mm,
we hope to do is send these. So we're going to be sending Russian bees and Pol-Lines over there because there are two stocks with a little bit different ways to keep Varroa levels low, just to see how that affects with tropy. You know, we're
thank you.
and so Jeff Williams's lab is the one to follow about tropolapse research. They're cranking out all kind of papers.
Is that the Auburn lab?
Yep.
Okay. They've put out some really good YouTube content on tropolalaps as well to educate beekeepers in the United States too.
Yeah,
Uh,
I collaborate with Jeff and his students a lot.
you guys are close together in geography down there in the
Oh yeah.
south. I want to cook through these questions to honor your time. I know you're an hour
Hey,
later.
no, I'll stay here as long as you keep asking questions.
Perfect. Eric asks a question. I have 28 hives in Denver. I select for grafting from my queen in a colony with low mite counts.
Mhm.
The daughter queens are open mated in an environment with many colonies. So he lives in like the Denver metro area,
Okay.
brought from across the US. Will my lack of drone breeding control mean I'm unlikely to improve resistance over time in my small stock of hives? And my response before you go into that is the more of us there are in the Denver metro area, Eric, the more of an impact we can make. So keep at what you're doing.
No, and that's exactly it. Is that yes, with drone non-resistant drone sources, if you're in an area where you don't have enough drones and they're being swamped out by other things, yes, it'll be you can make progress. Don't give up. Trust me. Keep going. It'll just take a little bit longer. What you really want to do is get all those people around you to also start raising some of these queens so they're producing the resistant drones. Because once you get those drones in place, oh, it becomes easy. And the reason why I say that is because that's exactly what we do at the lab. We kind of have an isolated mating yard where there's no commercial beekeepers and it's surrounded by basically suburbia. So we're pretty isolated, but we open mate a lot of our queens. Like I said, I'm producing like about 1,500 queens this year. There's no way to produce that many with instrumental insemination. So we're doing open matings, huge drone inundation areas. So typically, we start in January, right after New Year's, we start pumping those breeder colonies to be our drone sources. We start pumping them full of pollen, syrup, and giving them drone comb so they can start laying a lot of drones because we can produce all the queens we want, but we need those drones, and the open mated drones make it easy. So if you're interested, I would say you should probably talk to some of those neighbors and encourage them to use your queens. So there's market, get your stuff out there. But then also too, you have the benefit of having that area wide sort of cooperative of this trait being distributed at a high frequency. Now, the reason why I'm saying that would be very interesting is because that's exactly what we're doing with some local cooperators. So there's some here in Louisiana that's what we're doing with, where there's a cooperator network of probably like 30 beekeepers that everyone raises queens and they're all kind of in a kind of high density area. But what they're doing is they're grafting and everybody around there is getting resistant stock. And so we're going to be working with them to see how that level of VSH breeders rises in their population over time with that sort of cooperative program. We're also working with James Lee and the Sustainable Beekeeping Guild of Michigan. They're doing the same thing up there, trying to get everybody in that area to raise these queens so that way the drones do all the work for you. Yeah, drones, drones, drones. Need more drones. If you can, take those colonies to like some kind of isolated area if you can. But the other way to get around that is just try to encourage everybody to use your queens.
Frank, James, and both James Lee and Corey Stevens have been very, very generous with their knowledge and experience in helping us Colorado beekeepers. Those are two folks that I've reached out to. And that kind of leads to my next question before I hit Sue's. Um, I love how these people are kind of working from the ground up and trying to make this change, but this has been a hard thing from the industry is trying to make a change like Eric is talking about is how can the backyard beekeeper, the sideline beekeeper, even the vice president of a state out here in the West make a significant impact on the genetics that are being brought into a region or change the genetics. Maybe it's not bringing the genetics in, but changing the genetics. A sideliner business model of raising queens is a tough economic battle to pay the bills. Um, so it's hard from a sideliner level to provide those queens. I get how it's got to come from the commercial level, but it's hard to get good queens, and a lot of them that are really vetted and not just stated as VSH or mite resistant.
Because it turned Aristotle isn't giving this talk for the last 3 to 5 years. There's a lot more people making VSH queens, or uh, VHS queens I saw for sale.
Yeah.
So,
Yeah.
yes, it's out there. And yes, there are definitely some people who are doing the right thing, right? They're doing the breeding the right way. They're doing the evaluations the right way. They're swallowing their own, drinking their own Kool-Aid and going treatment free. There are some hardcore people out there like that. Then there's also people who said, well, I bought a breeder from Harbo 5 years ago. Well,
Yeah.
those aren't good queens. So what we're trying to do is come up with this. So an informed consumer, verified producer sort of thing. So that way, if you're a producer, you know what it takes to raise good VSH queens. So if you talk to Corey about this, Corey Stevens, uh, he's interested in kind of form this collaborative network, like any sort of producer of anything, right? You have kind of a group that kind of all has identities and standards and things like that. So that way the producers know how to actually do this. So they know if this is something they want to get into. Because this, you saw this presentation. This is hard work. This is not easy.
Right.
This is not the uh, magic bullet thing to all the beekeeping woes. It takes a lot of time and effort. So you have to be very, very invested. So knowing what it takes is really going to turn people off to it. And trust me, there's more people who are going to be interested, but then when they find out what it takes, say, nah, that's not for me. So I totally get it. So my rule of thumb is for every 50 people I give this talk to, hopefully one of them starts raising queens like this. So it's understandable. It's very hard. I mean, heck, we it's literally our job to make queens here at the lab, and we have a hard time keeping up with it. So that's us who are, we have every resource we need in terms of equipment and financial resources and just this colonies and land. We have everything, and it's hard for us. So I get it. Um, so that's one thing. Know what you're getting into. But then also too, from a consumer standpoint, which I think is the more important part, is to know what to look for because there are some less desirable actors out there um, that are just slapping a label on it and calling it resistant. So what I have here on this slide here is showing some of the questions that you need to know to ask people who are going to potentially be buying these queens from. So things like where did you get the genetics? Were they developed in-house or were they is this just Pol-Line that you're raising as your thing, which is fine. How are you doing your selection? Is this UBO testing? Is it Harbo test or is it mite washes? They're all going to have different outcomes potentially. Is your stock open mated or instrumentally inseminated? Right? Where are the drones coming from? How many drones are they mating with? How isolated is it? This answers back to that previous question about people in the area. More isolated the better, kind of thing. What's your treatment history? I mean, if you're still treating your colonies and you're trying to do breeding, it's going to be hard to know what's a treatment effect and what's a VSH effect. How many years of selection have you done? Because I wish didn't take 25 years to get to where we're at now, but this thing takes time. So you can see here there's a lot of other things that we're trying to work on, especially with the genetic identity. Ariane Avalos at our lab, he has a bunch of genetic markers that can pretty much predict VSH. So that's going to be a huge step forward. So there's all this is really come down to having a standard of identity and some kind of association standard in order to get this up off the ground. But what you could do if you're not a producer is just create demand because just like anything, where there's demand, someone will step up um, and start delivering this product. And I'll tell you what, the commercial queen producers, they know that this is the future because the reason why they didn't breed for resistance for such a long time is because amateur has worked. This is one of the most successful miticides ever developed. I mean, we had a 25-year run with it, which is unheard of in any sort of a pesticide sort of program because resistance develops and makes it useless. I mean, remember Apistan? That stuff that was used 20 something years ago? Why don't you use it anymore? Resistance developed. Kumafos or Checkmate, same thing. It went away real quick. We had a good run with Amatraz. It was good. It was good while it lasted, but we're at the end of that. We got to do something different. So they know that they could charge a premium for it if they could develop this, and they just need to know that demand is there, so they know how to shift their resources. So this is something that we're hoping to facilitate either from the supply side and consumer side.
No, when I got started as a beekeeper in 2014, the people that were standing in front of the rooms at the local clubs were um, discouraging the use of amatraz because of the history of synthetics and the tolerance that they saw coming down the line. So I've never used any of those products just because I was taught kind of from the beginning to go the organic treatment route if you were going to treat for mites. And I kind of see the wisdom of that in the past couple of years as amatraz has reached its end of usefulness. And I get it's got its role in the commercial industry and all, and it's an important role, but um, for people who don't run their bees like that, it's been helpful to not have to kind of rely on amatraz.
Yeah, and that's
Right.
something that is changing. I mean, now what there are other alternative treatments out there that are just as effective. We're finding out that just a couple years of non-amatraz application, you could restore that susceptibility in the mites. And so people are listening, and we're getting off the amatraz train. So that's good news. More products are being registered and developed, and there's so much research going into product development that we might not have new materials like new like chemicals, but like oxalic acid formulations are improving and things like
Yeah.
that. So I think that's actually in the short term, I think formulation of existing materials is going to be really important. But in the long term, we are going to need new miticides. And what's interesting is that honeybees, there is no miticide development pipeline at Bayer, for example, that because they're used to selling metric tons of active ingredient, whereas in the honeybee world, we might just use a couple thousand pounds, right, as the
Right.
whole industry. So it's not going to make the money, so they're not investing in it. So it's going to come up to um, startups that can develop these new materials and get them to market. What's really interesting, though, and this is a little bit of behind the scenes, is that EPA understands that the way that they regulate pesticides is geared towards row crop farming and not honeybees. So, like the registration costs are so high that no producer can afford registration costs if you're going to only make a couple $1,000s from selling this stuff. So they're understanding that, and they know that they have to change it specifically to honeybees because it's such a niche little tiny market.
That's good to hear that there's an awareness of that because I've actually had a couple of conversations with the folks who make the Easy Ox and
Oh
hearing
yeah,
them take a product that's already been tested, right, and proven as effective and jump through all those hoops. It's been tremendously difficult for them to do that. And hats off to people who will do that because it's almost disincentivized in a way to do it. So it's great to hear that the EPA recognizes that that
yeah,
could be improved a little bit. All right,
yeah.
Sue's got a question I want to make sure I get to. I
Okay.
have had Pol-Line Queen genetic and have had early and sustained buildup and a strong desire to build numerous queen cells over and over again, despite splitting and difficulty with queen acceptance at requeening. They have been great at surviving winter, having low mite counts, and producing honey. However, with multiple splits, I've gone through numerous frames of new foundation, one hive, split 4 times. Sounds like they're swarmy. Any suggestions on successful management of that trait? I'm beginning to think I need to manage these bees differently than I'm used to, like not encouraging early buildup by feeding, getting into hives more frequently, splitting earlier than usual. That brings up a kind of a similar thought that I have about the Pol-Line queens that have a lot of great traits. But here in Colorado, we have a lot of false springs. And that Italian-based genetics, from my experience as a stationary beekeeper, doesn't necessarily fit the temperate environments that we experience so frequently in the spring. And I wonder if that's a little bit of what Sue's describing in her experience here.
Yeah, and so we definitely see that too because sometimes so we open mate our queens, right? And
Yeah.
they're going to mate with about 20 drones. Well, maybe they might get 18 Pauline drones and then two other less desirable drones make it in there. And that could be what's happening because I think it's more of an isolated thing than a stock trait because we see that too is they'll build up quick and then they swarm very quickly sometimes. but those are outliers. I would probably say out of 100 colonies, we might see that, 5 times. So very low frequency, I would say, if that. It's Let me put it this way: it's frequent enough that we see it, but not anywhere near where we have to think. I think that it's a trait of the stock. And it's really important to let me just elaborate here a little bit more, is that when we're looking at this current version of Pol-Line, is that Garrett Dodds at our lab, who does all the breeding decisions, the first criteria he looks for is Varroa, right? Okay, it has to have low Varroa. But then he looks at honey production, gentleness, swarminess, all these things, and then makes his decision based on how those things are ranked. So he might have one that has zero mites, but it's very defensive. Nope, that's not a breeder. It might have zero mites, but it didn't make a lot of honey. Nope, it's out of it. So the current Pol-Line stock evaluation is pretty holistic in terms of desirable characteristics. So that's why we have, like, our breeder pool for the fall is usually like 300 colonies. And then out of that 300, we might find 5 or 6 or 8 that we actually breed from. So we're very, very picky on what we get. And so the other thing too is, if you're buying Pol-Line material from a different breeder, it can still be, Pol-Line base material, right? But it might be incorporating some things from the local environments that are potentially causing that problem. Um, some of it is regional characteristics. So, for example, we send hundreds of these around the country every year, and there are some places where they'll say, oh, when we try to install them, they didn't install. They either absconded or they superseded or very very quickly. Then there's other people who I send them to, and they'll say, I installed all 50, and every single one of them took. So there's some regional variations for whatever reason. Don't know what that trigger might be, but that could also be what's happening here. So that's about Pol-Line. However, what's interesting, and I think you brought this up with your climates, where you get those false springs, you said, or else
Yeah.
everything comes it next one day it's winter, next thing you know, 2 weeks later it's summer kind of thing. Well, it sounds like a carniolan type background would be really good for that kind of environment. And what's
Yes.
interesting is that there are some people who are trying to, well, no, not trying to, they have incorporated this trait into a carniolan background. It's not that hard. Just take it, made it to carniolans and back cross them, and you can incorporate that, because that's exactly right, is that some places they need a carny background, they need that quick rapid buildup. So maybe try a VSH in a carniolan background, and that might help you out.
Yeah. And again, it's that idea of supply and demand. There's definitely, and I hear you saying, and I heard Randy Oliver say it at the Bee Expo as well, at that roundtable talk you had, is, the consumer has to demand a different product for the producer to start producing a different product.
Right. And
And
and like I said, talking to the large scale commercial producers, they said flat out, we're not breeding for this because people treat, and treatments work. Well, I said,
yeah,
well, what if the treatments don't work? Well, then we'll have
yeah,
to revisit this. So that's kind of where we're at right now. So yeah, like I said, demand is there.
right on. Thank you so much for your time, Dr. Rinkevich. I see how late we are going. If people want to reach out and contact you, how might they do so?
Best way is
I,
through email.
I knew you were going to have a slide for it. Perfect.
Yeah. Yeah.
Right
Um,
on. And then what's going on in your B lab in the next couple months? Where will you be? Are you going to any big conferences and speaking in the future? What's coming up for you?
so this summer, bad part is people lost a lot of bees last year. Good news is
Yeah.
we have a lot of funding. So I have a lot of money that I have for research this year. So from July 14th through August 29th, I'll be on the road. I should have like a rock and roll T-shirt that says the Rinkevich Research World Tour.
With a tour, with all your
Yeah, yeah, yeah.
stops on the
Coming
back.
to a city near you. Yeah, so we'll be going up to Michigan, spending like 2 weeks in Michigan, then 2 weeks in North Dakota, then 2 weeks in Montana. Come home, go to a wedding, then after that wedding, go down to Florida and Texas. So we're gonna be all over the country, uh, doing this research of Amitraz resistance, evaluating bees for VSH and making breeding decisions. That's pretty much gonna be my July, August, September. So the only thing that's in between there is a friend's weddings.
And
Hey,
is Rogan Tokash still with you, helping out with that work?
well, Rogan got a job at Fargo up in North Dakota,
Oh,
and he starts,
okay,
I think, in 2 weeks. So,
okay,
oh no, a little his defense is in 2 weeks. Yeah, so I'm gonna be collaborating with him up there. That's what's kind of
awesome.
nice is that I'll be able to have like a home base up in Fargo with him being up there now. Excited.
Yeah, exactly. And then are you speaking anywhere coming up as you do the winter speaking conference route tour? Is
I hope to.
that too far down the line?
I'll tell you about it when we come to it. Hopefully, you'll see me at a
Yeah,
meeting this fall or
yeah,
winter.
sounds good.
Yeah.
Right on. Well, we appreciate all the work that you do at your lab down there, and your lab, not just you, but all the people down there at that lab do. Keep up the good work, and we love to keep kind of in touch with you to see how things are progressing as you continue on your work down there.
Well, thank you very much for inviting me today because this is I always, these are good questions, and this really shows that beekeepers are looking to the future. And I think this is going to be a very big part of our beekeeping future. And I'm just saying that, and I'm not just saying that because it's my research, but I think that, this is what the data are pointing us to, right?
Yeah, yep,
And except when I talk to beekeepers who use this stock, and I said, what were your losses like this year? They said, oh, 15, 20%. So, I mean, I
yeah.
know this is really important, and it's really good to see that there's interested people out there. So thank you very much for inviting me, and I'd be more than happy to give you a talk about this research or anything else that we're doing. It's always good to have informed beekeepers. So thank you very much, and you have my email there, so send me an email, and we'll keep the conversation going.
Will do. Appreciate it. Thanks a lot for all your
All right,
time
good night,
tonight,
everybody.
Dr.
Thanks for having
Rinkevich.
me.
Thank you,
Have a
Dr.
great night.
Rinkevich.
All right, good night. Bye bye.
Night.